Muscles represent strength, and the history of mankind is peppered full of muscular heroes performing unusual feats. Considered as a sign of virility and strength, muscles are visible beneath the skin. Whether literally or figuratively, the muscle is central to all human action, its contractile role always in keeping with the required gesture. However, a muscle is never mobilized in isolation. There is a harmony that spreads from one muscle to the next during movement. One wonders then if this harmony is entirely orchestrated by the central nervous system, or whether it also proceeds by way of an inherent elasticity that spreads from muscle to muscle. To find out, we shall explore the muscle within its environment in a living person. The paravertebral region surrounding the insertion of the latissimus dorsi muscle with the muscle layers of the back and the rib cage is a perfect area to explore. The muscles are just below the skin, in direct continuity with the hypodermis and the vertebrae. The latissimus dorsi and paravertebral muscles are strong and imposing structures in the thorax which facilitates their anatomical identification. These macroscopic observations cannot ignore large areas where sliding occurs, permitting movement between muscles as well as between skin and rib cage. As the camera slowly approaches the area between distinct muscle groups, the observer will be impressed by a genuine intertwining and interweaving of opalescent fibers, which create a link of total continuity between the muscles. These fibrous images transform into images of sparkling, scintillating, mobile mirrors and ephemeral lights, disappearing instantly, only to be replaced by others. What are these reflections? Why do they exist between muscular structures? So, in order to progress further, the observer will be required to separate, lacerate this amalgam of structures which do not seem to be part of the organs and look negligible. In order to expose the structures, as during surgery, one must break through these clusters of dense, heterogeneous and continuous fibers, which give the impression of surrounding and wrapping around all elements, binding them together. This veritable continuum of fibers, which is present in all spaces, has been named connective tissue by the anatomists. It's present everywhere, from the muscular depths to the surface of the skin, connecting separate elements. To the naked eye, this tissue at first seems fairly uniform and unimportant. The mere introduction of an endoscope during surgery changes everything. The endoscope combines a high-definition camera with a coherent bundle of flexible glass fibers, a lens, and a source of cold light. Thanks to this new technology, it is now possible to obtain degrees of definition never before achieved. The diameters of the lenses are either 4 mm or 2.5 mm, with excellent focusing but with limited depth of field. Added to this is the problem of having to constantly refocus, as well as the strange apparition of mist, droplets and fatty deposits that require constant cleaning of the lens. Endoscopy allows us to enter an arachnoid world of extreme morphological diversity and, without prior knowledge, discover an unexpected biological structural organization, opening up an abyss of meditation. This apparent monotony contains a range of diverse colors and forms that are almost geometric. Moving the high-power lens into the areas surrounding the muscle allows us to discover the blood vessels that supply the energy, clusters of cells in plates curiously spread over the surface of the muscle, or within the sliding fibers. Again, we find the spaces filled with scintillating facets, refracting the light like mirrors dumped haphazardly in a heap. Indeed, the cold light admitted by the endoscope enhances certain contrasts, and it's easy to observe that the mirrors themselves consist of interwoven fibers associated with liquids disposed randomly throughout the spaces. Here, the opalescent veil is woven from small cables and chaotic bundles. We have seen in the previous films, Strolling Under the Skin and The Skin Excursion, that these fibres consist of collagen, especially type 1, and contains liquids such as glycoaminoglycans with varying chemical compositions. We also know that these fibres are irregularly disposed, apparently fractal, meaning that the large fibres divide into smaller ones, which in turn divide into even smaller ones themselves. These fibres and liquids define the spaces that we have named microvacuoles. The formation by these dispersed fibres of a homogeneous entity define what we call the connective tissue. But does this also apply to muscle? Could it be that we also discover a fractal organisation that would, for example, provide a link between the muscle fibres, the epimysium, perimysium, and the endomysium? We would then be in the presence of a coherent, global and functional tissue, rather than distinct histological entities. Once we have cut through the skin, the muscle is not far. It is distinguished by its purple colour and stringy texture, and contracts as soon as we use the electric scalpel. The muscle is not, however, an entirely distinct entity from the surrounding anatomical structures, because the muscle is surrounded by this connective tissue, and it is necessary to peel away the epimysium to really get into contact with the muscular structures. Sometimes the epimysium of certain muscles is surrounded by a reinforced membrane which permits sliding during contraction. This is the aponeurosis. Sometimes the sliding system is more tenuous. Entering a muscle is not easy. There is a strong resistance to the endoscope by what feels like braiding of the fibres. Even though the show is amazing, the scientific harvest is poor and one is forced to retreat. The endoscope is orientated towards areas of easy exploration. We let it slide towards these opalescent short or long fibrils and blood vessels of varying forms in the areas surrounding the muscles. This is what we call the epimysium. Epimysial fibres are in continuity with the surface of the skin and blend into the hypodermis, as we have already described in our previous film, The Skin Excursion. But they also enter, leave, mix with, combine and penetrate deep into the perimysium, separating the bundles of muscular fibres. It seems obvious, apart from the sheer beauty, which is not negligible, that everything is connected. There is no break in continuity of living matter. There are no sheets of tissue, layers or sublayers arising from nowhere. The epimysium and perimysium are continuous structures. As for the endomysium, which binds the muscle cells together, its finesse means that we can only guess at its presence during endoscopy. The projection of the fibres of the perimysium seems to be much more disorganized and global. Compared to the longitudinal and parallel aspect of the muscle cells, their architecture is neither parallel nor regular. These perimysial fibres surround, penetrate and encircle the bundles of muscular cells. However, we have the impression of tissue fusion between the collagen fibres and the cells which compose the bundles, while at the same time preserving an apparent anatomical independence. The muscle cell nestles within the fibrillar architecture in the same way as the adipocyte nestles within the fatty lobule. One might think that the collagen fibres and their finer components mould and shape muscle cells by way of their intimate architecture. And given that these endless or more or less diffuse networks of collagen fibres and fibrils link the large and thick connective formations, such as fascia and tendons, this raises the real question for those who work with living organisms. Is this connective tissue not simply the tissue that is ignored by all manuals of anatomy and histology? Could it play other roles than that of a simple supporting tissue, which is the accepted term, but whose meaning is still rather vague? Despite fine focusing and high magnification, sometimes as much as 50 times, providing high-definition images, endoscopic exploration does not enable us to see the ultimate subtlety of detail. The absence of contrast does not permit us to go much further, and so the doubt about continuity between all these structures remains, particularly at the level of endomysium. The use of other techniques can therefore be valuable in defining the boundaries and exact nature of this system. To achieve this, we will use the electron scanning microscope on a rat's paw. The rat's paw has been treated with formalin and prepared using a technique called sodium cast. This entails soaking the rat's paw in a solution of sodium hydroxide. The paw is then rinsed and freeze-dried. What remains is what you can see here, white tissue in which all muscular and cellular components have been eliminated. However, the electron scanning microscope only shows the surface of objects under investigation. One must then penetrate into the interior of the paw to be sure of what we are looking at. We then proceed through a binocular magnifying glass. The electron microscope enables observation of a greater depth of field, practically within the space under observation. As a result, one can observe the global structure of the object under observation, in this case the rat's paw, at low degrees of magnification, so enabling us to observe the finest details of the finest fibres of connective tissue.
Et observer les détails les plus fins, les fibres les plus fines du tissu conjonctif.
This allows us, as shown in this image at low magnification, to see the remains of the gastrocnemius muscle and to see that the collagen fibres present between the gastrocnemius and the extensor digitorum join up with the epimysium of the gastrocnemius. Then if we move into the muscle at higher magnification, one can see that the collagen network that constitutes the epimysium continues into the muscle via a network of parallel and oblique structures. Then collagen fibres ramify into the formed space to a scale equivalent to the size of a cell, which is about 50 microns. All these collagen structures within the muscle link up with the other part of the muscle, which happens to be the tendon, easily recognizable by its tightly packed parallel bundles of collagen fibres. The collagen fibres of the tendon branch out into turn to form the paratendon, branching out again into a very fine network of fibres, which interweave with the sliding aponeurosis and further afield with the hypodermis.
What are the implications of the microscopic and endoscopic observation? The essential observation is of fibrillar continuity. There is no break in tissue continuity, be it with muscle, tendon, or around the arterial and venous structure, and also the structure surrounding adipocyte. All these structures are formed in the same manner and are continuous. We have already encountered these problems when we discussed the subcutaneous tissue intruding under the skin and the epidermis and dermis in the skin excursion. Now we have discovered the same continuity of the tissue within the muscle. The concept of the organization of the living matter into stratified layers, hierarchical layers of sheaths, lamella and strata, cannot satisfy an anatomist who studies precise endoscopic and functional anatomy. It is certainly useful in order to learn simplified and descriptive anatomy, but it is the fundamental concept of the global nature and continuity of tissue that one must now accept. Even though many of different colors, texture and shapes, they are all linked to each other. This is a global tissue concept. We have already demonstrated and highlighted these fibrous structures with irregular three-dimensional organization that can be considered as chaotic. Perhaps at the muscular level, we can now envisage the same concept and consider that muscle is an assembly of interwoven collagen fibers oriented in the direction of contractions and cellularized by muscle fibers, which are the contractile component, within which, of course, everything is interlinked. This view leads to conceptual consequences. The first of these concerns the creation and transmission of force, which is, of course, the principal role of muscular function.
Indeed, if these elements are linked, all mechanical stress will act on all the muscular structures, whether they are epi, peri or endomysial. But how and where does this transmission take place? We can clearly see in this sequence that traction on a muscle spindle induces a transmission of the force to the neighbouring spindle, and that the collagen tissue participates. Seen from afar, there is nothing mysterious about this, but if we look closer, the behaviour of the fibres is surprising and far from behaving simply like ropes. During careful endoscopic observation, we have found that these perimycial fibres move over each other. But this is not a regular or direct deployment of the fibres. Instead, we can identify a series of simultaneous, unpredictable movements, such as division, lengthening and sliding of fibres. Watch these collagen fibres with a diameter of 20 microns in the perimycium. Within the space of 2 seconds, we can see mobility, division and distension. This is surprising, but reinforces our hypothesis of global behaviour, because all these movements have already been described beneath the skin, around the tendons, and now we find them around and within the muscle. These collagen fibrils possess their own nonlinear dynamic relationships, but how are they attached to the contractile element, the muscle cell? In vivo, endoscopic observation does not identify the exact nature of these musculo-connective tissue links. Here again, electron microscope observation after freeze-drying tissue specimens, that is after setting all the components of the muscle and not just the collagen, will enable us to reveal the existence of physical links. When we observe the surface of a muscle cell at high magnification, we can see collagen fibres spreading out from a strand of perimycium like ivy branches, and then coming into contact with the surface of the cell and the endomycium. And even more amazing, in this picture, we can clearly see that the collagen fibres are continuous, with the structures contained within the membrane of the muscle cell, and that they are even continuous with the cytoskeleton. So, in this way, the contractile forces produced within the myofibril can be transferred both morphologically and mechanically to the tendons, especially since we find these links at regular intervals of about 300 microns along the muscle cells in all 3 dimensions. In reality, each muscle cell is closely associated with the collagen network via crosslinks within an endomycial framework, composed mainly of type 4 collagen fibres, which connect the muscle cells to each other over long distances. Trotter and Purslow have demonstrated that the force produced by a contracting muscle cell can be transmitted laterally through the network to its neighbours. So, what happens during muscle contraction triggered by a nervous impulse? The contraction causes the intermingling of the actin and myosin fibrils located in the sarcomere. This contraction then spreads through the cell towards the tendon, but it also spreads laterally to the endomycium and especially to the collagen fibre bundles within the perimycium. The movement is subsequently transmitted to the large collagen bundles that merge with the tendon. However, the collagen bundles do not all attach to the tendon. They also constitute the epimycium, which represents a link between muscles and which could transmit some of the force to neighbouring muscles, as demonstrated by Oying and al. Finally, the muscular contraction is transmitted by a dissemination of forces via this 3-dimensional network, which links together all parts of a muscle. This transmission and modulation of force throughout the entire structure is of great interest because it helps to explain why blood vessels don't rupture during muscle contraction when the energy demand is greatest, thus preventing bleeding and bruising. One also wonders whether the connections between the perimycium and the muscle cell are formed randomly along the cell surface or at specific locations. In order to find out more, we needed to go back to the laboratory, and here the transmission electron microscope provided the answer. This time, we used a transmission electron microscope. We took a small piece of muscle, which was prepared using special techniques. It was then impregnated with and set in an epoxy resin, which is very hard and allowed us to cut very thin sections of the prepared tissue. Subsequently, the electronic microscope allowed us to observe and learn what was contained in the prepared section of tissue. This longitudinal section of a contracted muscle cell from a rat will provide us with the initial indications. Here at the top of the picture, in contact with the perimysium, the cell is slightly deformed, and we find one or more nuclei and impressive clusters of mitochondria. The same observation can be made in a cross-section of 2 muscle cells from a bull, showing that the type 1 collagen fibres of the perimysium, identified by their striation, establish contact at places where there is a nucleus and abundant mitochondria at the periphery. Such coincidences cannot be due to chance alone, since we know that the nuclei control the production and maintenance of proteins, and that the mitochondria are responsible for the metabolism of energy and partly for calcium. A phenomenon of mechotransduction can therefore be suggested. This global conception of muscle as a zone of contractile cellularization of collagen tissue, which provides architecture and form, with links to all other tissues throughout the body, is reinforced by this last sequence filmed beneath the skin. Here we see that manipulation of the surface of the skin triggers multidirectional movement of the fibers of the muscular epimysium beneath the skin, demonstrating a direct physical interaction. Perhaps research into myopathy will make use of these new avenues of exploration. However, it is certain that the benefits provided by massage, manual therapy, or simple regular physical exercise can now be explained in a rational, physiological, and non-controversial manner by considering their effect on the flexibility of these contractile and connective structures that are so intimately linked.